So the purpose of our study was to identify an assay or a combination of antibody biomarkers that could distinguish people who have been recently infected versus long-standing infections. And this is really important for the field because in 2016 there were 1.8 million new infections. The field is trying to improve upon prevention modalities, but in order to understand if these prevention modalities are effective, we really need to understand the number of new infections per year. So a number of assays have been developed that have looked at HIV incidence. We've improved upon these assays by being able to decrease the false recent rate in those assays, as well as be able to determine recent versus long-standing infection in the presence of antiretroviral treatment. So the currently available assays are not accurately estimating HIV incidence. They don't have an appropriate window for identifying recent from long-standing infection. They also have a relatively high false recent rate. So they're misclassifying people as long-standing or recent infections. So additional problem with the current assays is accurately classifying people who are taking ART or antiretroviral treatment. Especially with new prevention modalities such as treatment during acute infection as well as pre-exposure prophylaxis or PrEP. There will be people who have ART circulating in their blood, and the current tests need to accurately identify recent versus long-standing infection in the presence of ART treatment. So we performed a retrospective analysis of HIV-infected patients over time to profile their circulating antibody response to the HIV virus. We began this work when we were looking at acute versus chronic infection and noticed a really striking finding that antibody responses to infection appeared sequentially. So the person's immune response recognized the HIV virus, different parts of the HIV virus sequentially, as well as different antibody forms came up at different time points. And we also noted that we can identify the kinetics of those antibody responses coming up that are recognizing the virus and the maturation of those antibody responses over time. And so the methods that we used harnessed these early findings at looking at the immune response to HIV infection and developed a binding assay that could look at the reactivity of the antibodies to different regions of the HIV virus, the strength of that interaction with those different regions, as well as the maturation of that response over time in terms of different forms of antibodies. And then we used a computational analysis or machine learning to pull all of those different measurements together to try to identify a combination of biomarkers that could accurately measure and distinguish recently infected versus long-standing.
The improvements seen with this set of biomarkers are a tremendous benefit to the field because they allow for public health researchers and epidemiologists and those treating HIV to more effectively target prevention and treatment strategies by having an accurate handle on the epidemic and where hotspots are occurring and who's becoming recently infected versus who's been infected for a long time. And again, these improvements are mostly seen in those participants who are already on antiretroviral therapy. The research has progressed to the point now where we are ready to begin development of a kit which would put this into the field in the hands of people who could measure incidence at regional, state, and national levels.